Good day, viewers. This is 300 Plus Academy, where all we do is exams tutoring, ensuring that you ace your next exams. I am Adeyemi Abiodun. So, we'll be looking at the subject of life today. Having looked at cytology, looking at cells, uh, taking a tour of eukaryotic cells, looking at forms in which cells exist, organization of life, and of course, variety of cells. It's about time for us to look at how substances move into the cells and how substances move out of the cell. That is why we said movement in and out of cells, or what we call transport across cell surface membrane. Of course, this is the cell membrane, or what we call the cell surface membrane. So how will material move across the cell surface membrane? And since the cells are in our body fluid, you look at the body fluid, they contain ions, they contain materials when we eat, and food particles are broken down into soluble food. Take, for example, carbohydrates broken down into glucose, proteins broken down into amino acids, and these soluble compounds are in the body fluid, they are in the plasma of the blood precisely. From there, how do they then get into the cell? So we say cell and its environment. The environment of the cell is the fluid surrounding the cell. If it were to be a unicellular organism, then you talk about the water in which that unicellular organism lives. But the important thing is this: materials must move in and out of the cell. And for us to understand how that happen, how materials will be able to breach this wall. I call this more of a wall, more of a barrier. Now, how will materials be able to move in and move out? Because this is more of a barrier. Now, look at it. I said the cell membranes present barriers to the movement of ions and molecules particularly. I think at this point it's very important so that I let us, know about the real structure of the cell membrane. When you talk about the cell membrane is not a line as most of you have been made to believe. At a much higher level of things, via what we call the fluid mosaic model, or the trilaminar nature of the cell membrane. When you talk about the cell membrane is a phospholipid bilayer. Here is the phosphate, here is a lipid, here is a lipid, here is the phosphate. Now, the thing is this: we usually don't have problem about the phosphate because they are polar. In other words, they are hydrophilic, more or less in layman language, water loving. Recall that when you say water, H2O, water is a polar solvent. Water is polar. So the thing is, when you look at something like glucose 6-H12O6, when we draw the structure, it contains oxygen and hydrogen. So definitely glucose is polar. Same for amino acids. They are polar. So that's their hydrophilic. So that is the nature of these chemicals. This from carbohydrate, this from proteins. They must get into the cell. Now the issue is that when you talk about the lipid part of the membrane, it is a hydrophobic region. It is a non-polar region. It is not charged. So it more or less repels and won't allow anything that is polar to pass across. Look at it. Lipid. These are hydrophobic or non-polar region. It will never allow glucose, hydroxyl of glucose that makes glucose charged, amino acids. It will never allow them to pass. You talk of something like cholesterol now. The lipid part is going to allow them to pass. As you see on your screen, look at the trilaminar nature of the cell membrane. You can see that we have phosphate and the phosphate tail. Now, you can see the carbohydrates on the outside. They cannot enter. They are on the outside for now. Something has to carry them across. You look at the purple transmembrane protein. That is what the carbohydrates will attach to get them across. you look at the protein towards the end. That is exactly, even protein will attach to what we call carriers on the cell membrane. But when you look at cholesterol, you can see it's getting in between the phospholipid bilayer. You can see it in between the lipid because it will allow that to pass. You can see cholesterol will pass because it is also hydrophobic. It's going to pass. So the real issue is usually the lipid portion. It repels this and this and won't allow them to pass. So what then carries this into the cells are what we call membrane proteins or carriers. That is what carries things like protein or amino acids into the cell. When you talk of things like oxygen, water molecules, carbon dioxide, these are smaller molecules. They could find their way through tiny spaces and get into the cell. There are usually no problem with this. The problems are actually with the polar molecules like glucose and amino acids. The lipid presents a barrier to them, and that's why I said cell membranes present barriers to the movement of ions and molecules, particularly polar molecules, that's water-soluble molecules such as glucose and amino acids that are repelled by the non-polar hydrophobic lipid of the membrane. So that's the problem. But that notwithstanding, transport of materials must still occur. Look at that diagram again. You can see this is your phosphate. This is your phosphate. We usually don't have problem about the phosphate. The real issue is with the lipid, and because that's simply because they are hydrophobic, that they are non-polar, and meanwhile these materials here are polar, so there is no way they are going to get access. They would get access via membrane proteins and carriers carrying them across the cell. We are going to talk more about this later, but when you look at the nature of the cell membrane, something is already clear, that the cell membrane is a living layer, one. That secondly, the cell membrane is selectively permeable. It will allow some substance to pass through. It will not allow some substance to pass through. It will give some substances like oxygen, water, carbon dioxide more of a free passage. So that's why we talk about the semi-permeable nature of the cell membrane. We talk more about that when we get to osmosis, but the thing is this: for us to understand how materials move across the cell surface membrane, we need to first of all understand certain things about the cell membrane, and that is why we are talking about the trilaminar nature of the cell membrane. That notwithstanding, transport across cell surface membrane must still occur. So you can, that's why the topic is movement in and out of cells, because the right materials must move into the cells, secretions must move out of the cells, waste materials or unwanted materials must move out of the cells. There must be ionic balance between the outside and the inside of the cells as well. So you're talking about movement of ions as well. And since most of these molecules, most of these materials are in the environs of the cell. In the case of when we talk environs of the cell, take humans, for example. The environs of myself, for example, is the plasma. When I eat and food is being broken down and digested and converted into soluble foods, they will be in the plasma of my blood. From the plasma, they then from there they move into the cells. When materials are released from the cells, they'll be released to the plasma. When you talk about unicellular organisms, you're talking about, most unicellular organisms are aquatic. You're talking about the water surrounding them. It is from that water that dissolved oxygen will move into the cell. Carbon dioxide will move from the cell outside. They get their nutrients from that water. So that is why for materials moving in and out of the cell, you're trying to look at the cell in relation to its environment. So you can as well call this topic cell and its environment. But take note, we've talked about the cell membrane. Whatever be the case, transport across cell membranes must still occur. for a number of reasons. The first and most important is to obtain nutrients. Secondly, to excrete waste materials. Thirdly, to generate the ionic gradient essential for muscular and nervous activity. We're going to talk about sodium, potassium pump when we get, of course, to the action potential of the cells much later when we discuss things like muscles and nervous system. Also, transport must occur to maintain suitable pH and ionic concentration within the cell for enzyme activities. But even if you don't get what this two is about for now, at least you know about this two. The right nutrients must move into the cell, talking about glucose, amino acids, fatty acids, and glycerol. Oxygen must, respiratory gases, oxygen must move into the cell. Carbon dioxide must be eliminated, and the likes. So our focus now is going to be how to maintain the life in the cell via the right materials getting in and wrong materials getting out, secretions moving out as well. So that said, now looking at the processes or mechanism by which substances move in and out of the cell, there are four basic processes. The first one is diffusion, which deals with molecules, oxygen, carbon dioxide, and the likes. The second one is osmosis, which deals with movement of water, active transport, then what we call bulk transport, that's talking endocytosis. In the case of endocytosis, it simply means the cell is taking in the materials, and when it is taking in things that are solid, let me put it that way for now, you're talking phagocytosis, otherwise known as cell eating. In the case of liquid soluble particles, molecules, you're talking pinocytosis, cell drinking, in other words. And then exocytosis, in this case, the cell is exporting material. So there are four basic mechanisms: diffusion, osmosis, active transport, then what we call bulk transport. But it might interest you to note that this classification is based on energy requirement. It is based on if the cell will use its own ATP, if the cell will use its own energy to carry out the processes. Processes that does not require the cell using energy to carry it out are called passive processes. You see, it is you carry out this process without use of cellular energy. No ATP is consumed. So when I say passive transport or passive processes, it simply means transport is occurring here, but the cell is not having to expend energy. So when you talk about diffusion and osmosis, they are passive process. But when you talk about active transport and then bulk transport, they are active process in that the cell will now use its own energy, its own ATP to carry out the process. So this classification is based on energy requirement. It's based on if the cell will use energy or not in carrying out the process. So recall diffusion, osmosis, they are passive. Active transport, bulk transport are active processes. Then there is another classification which is based on, recall that we talked about the nature of the cell membrane. It allows some substance to pass freely. It will not allow some substance, especially the lipid layer, the lipid region of the cell membrane. So those substances that are not allowed, that are hydrophilic, that are not allowed in the non-polar hydrophobic region of the cell membrane, talking about the lipid, now have to depend on carriers and transmembrane protein to carrying them in and out of the cell. So based on what carries the molecules into the cell, the mechanisms are classified into three. Diffusion and osmosis, they do not need carrier. Then we have those processes that has to be mediated by membrane proteins or carriers. It is membrane proteins or carriers that have to carry those particles across. As you can see on your screen, you see carbohydrates, being attached to the membrane protein and the carriers there so that they carry them across. So those ones, we call them mediated transport, in that transmembrane protein and carriers now have to interfere. Substances bind to proteins or carriers on the membrane to carry out their processes, to carry out their to carry them, sorry. Substances bind to proteins or carriers on the membrane to carry them across. Example of mediated transport are facilitated diffusion, which I'm going to talk about much later. Yes, facilitated diffusion, and then active transports. These are mediated transport in that membrane proteins and carriers are what we now have to carry the molecules across. And thirdly, we have bulk transport, talking about endocytosis, cell intake. If the intake is solid materials, you're talking phagocytosis. If the intake is liquid materials, you're talking pinocytosis. And then exocytosis, of course, exporting materials from the cell. So these are the classification of the processes whereby substances moving in and out of the cell. This is based on energy requirement. This is based on if a carrier is needed to make the process occur. So what we are now going to do is we have to now take each of them one by one, discuss them, look at how it occurs in organism. When we say diffusion, let me have a cube sugar, a lump of sugar, as we see in the diagram on display. You can see the moment we drop the lump of sugar in water, you can see how the particles of sugar there begins to spread. So the very spot where the sugar are dropped, we say is having a high concentration of sugar particles there. And then it suddenly begins to move from that spot to other spot. It begins to spread evenly. In other words, we say it begins to move from region of high concentration to region of low concentration. When one region has a high concentration of certain molecules and the other region has a low concentration, we say it is moving down concentration gradient. We say it is moving downhill. We say it's moving from a region of higher concentration to a region of lower concentration, as you can see in that diagram. Then you see that the particles there begins to spread evenly until it spreads all across the water. This is what diffusion is about. Diffusion is about spread. If you look at this situation now, it's a case of solid particles, solid molecules being distributed or conveyed by liquid. So the medium conveying here is liquid. So you're looking at spread of solid via liquid from a region of high concentration to a region of lower concentration. How about if we drop liquid ink in water, or let's say dye in water? The very spot where the dye are dropped, is having a high concentration, as you can see on your screen, and then it begins to spread to other region. It begins to spread. This is liquid ink now. So this is a case of liquid being spread by another liquid. But the molecules, the particles of the dye, the green dye, as you can see on your screen, is moving from a region of high concentration to a region of lower concentration. And the medium that conveys that particle or those molecules here is liquid until it is evenly spread all through the liquid. This is another clear case of diffusion. Or let's assume we have a dead rat somewhere in the corner of the room, as seen in my college biology, as read in my college biology. Let's assume we have a dead rat at the corner of the room. It's been dead for like 3 days. What we notice is that the very spot where the rat has died, the odor, the offensive odor of the dead rat is going to be more intense or pronounced, and then it gradually begins to spread from that region of high concentration to other region in the room, or to other region in that apartment. So you see the odor, that's the gaseous molecules, are moving from region of high concentration to region of low concentration, and what conveys that odor now is air. So the medium in this case is air. Or let's use something palatable. let's assume the odor coming from the kitchen of a busy housewife, exactly how it's been put in college biology, coming from the kitchen, the odor, the sweet savour coming from the kitchen of a busy feminine housewife, let's say making jollof rice, Nigerian party jollof, that begins to spread from the kitchen at the very spot where the meal is being made, where the jollof rice is being made. The odor is a lot more intense and then begins to spread gradually to the sitting room, to the dining, to the sitting room, and then gradually to the outside, such that one can, can perceive the odor. That is movement of gaseous molecules from region of high concentration from the kitchen to the outside, low concentration, and the medium that carries now is air. So that's what diffusion is about. Diffusion is about spread once there is a concentration gradient. Once you have a concentration gradient, diffusion is going to occur. To say concentration gradient means one region having a high concentration, the other region having a low concentration, then diffusion is going to take place. In other words, diffusion will continue until a stage is reached where the molecules are well evenly spread. We say equilibrium is reached. Example is when you drop solid KMNO4, or as in the case of sugar, which I've shown you, in water, then it begins to spread. Dead rat in a room, the odor continues to spread. Liquid ink spread in water. So once there is a concentration gradient whereby, once there is a concentration gradient whereby one region has a high concentration, another region has a low concentration, diffusion is going to occur downhill. That's downhill here means from high concentration to low concentration to spread around or distribute substances uniformly. And you need to know that when you talk about diffusion, let's take, for example, outside me, there is high concentration of oxygen. On my lungs, there is low concentration of oxygen. Okay, let's say this is my trachea to my bronchi. Of course, let this be my lungs. On the outside here, there is high concentration of oxygen. Oxygen is high. On the lungs, there is low concentration. So oxygen will move from region of high concentration to region of low concentration. Now the oxygen is here. When blood flows to my lungs via the pulmonary artery, now there is high concentration of oxygen from the lungs. There is low concentration of oxygen in the blood. Oxygen should therefore move into the blood. Now, the blood coming to the lungs is blood, carbonated blood, that's deoxygenated blood. It is having high concentration of carbon dioxide, and now there is low concentration of carbon dioxide on the lungs. Carbon dioxide will move to the lungs. So you can see that exchange of respiratory gases here is occurring via diffusion. Once there is a concentration gradient, diffusion is going to proceed. Another carbon dioxide is on the lungs. On the outside, there is low concentration of carbon dioxide. Carbon dioxide will move from the lungs, boom, to the outside. So that is how that process occurs. So you're looking at diffusion occurring in living things here. And if you look at the examples I've used, dropping sugar in water, dropping solid KMNO4 in water, dropping 1 or 2 drops of ink in water, the odor from the kitchen, dead rat. These are non-living. Now this is living. If you talk about a plant, during the process of photosynthesis, carbon dioxide is needed inside the plant. So carbon dioxide from the outside will move into the plant via diffusion. There is oxygen is released as a byproduct during photosynthesis. The oxygen will move out by diffusion. So you can see that when you talk about diffusion, it occurs in both living and non-living things. And there's something about diffusion. Diffusion occurs in both directions. So long as there is high concentration here, there is low concentration here, diffusion will proceed. If there is high concentration here, there is low here, diffusion will proceed. So diffusion occurs in both directions. Take note of that. So moving on now, the next thing is to talk about the factors that affect the rate of diffusion, factors that determine how molecules or ions spread across the cell surface membrane. Of course, the first one is the steepness. It's all called the steepness of the concentration or diffusion gradient. That is, we're looking at the difference in concentration between point A and B. The more the difference, the faster the rate of diffusion. That's the steeper the concentration gradient. Let me use this mathematically as though we want to calculate slope in mathematics. Okay, imagine I have let this be point let this be point A, let this be point B on my graph. Okay, so that I talk of this is going to be my slope. So that my slope here is going to be, this is going to be time. So let the change here, let it be, let's say from here to here, let it be 50, and let this to this place, let this be 5. So you see the slope here is going to be, the gradient or the slope here is going to be 50 over 5. That makes it 10. Now let me have another graph this way. This is this way. And let this be my point A, let this be my point B, so that the change here, let this change here, let this be 20. You can see from here to here 50, from here to here 10. And from here to here is 5, from here to here, let it be 10. So that if I say 20 over 10, my slope is going to be 2. My slope here is going to be 10. And you see, this is a lot more steeper than this. This is steeper than this. In this case, look at the way the slope is here. Look at the way the slope is here. It has been reduced. So the thing is this. We can bring something like this thinking to how substances spread. The more the concentration difference between 2 points, the faster the rate of diffusion. Picture a case, let's use numbers now, where the concentration difference between point A and point B is 1000. Let's point A have 1000 molecules, let point B have 0 molecules. Now, when molecules are going to spread, they begin to spread, let's say in 50s. 50 molecules move from A to B, and then another 50. But picture a situation where the difference between our 2 points is just about 70. If molecules will move from point A, which has 70 molecules, to point B, which has 0 molecules, then the particles here is going to move in probably 5s or 10s, not in 50s or 100s. So the more the concentration between 2 points, the faster the rate of diffusion. In other words, we call that the steepness of the diffusion gradient or the concentration gradient. That's what determines the rate of diffusion. Another thing is surface area. You see, the more the surface area of a membrane, the greater the diffusion rate. The larger a cell, the smaller its surface area. And this can very well be indicated. If you have a cell of this size and a cell of this size, this cell A is going to be a lot more effective than cell B. Very easy. Materials or molecules get to the center of the cells very quickly, other than getting here. It takes a longer time getting to the center of the cell. This I talked about while I was talking about cell biology. I asked the question, I said, why cells are small? Cells are small just for effectiveness reason. In other words, for communication reasons. When we say communication here, we mean the rate at which materials get to the center of the cells very quickly. That's what we're looking at. For this cell A, materials will get to the center very quickly than cell B. So cell A is going to be a lot more effective than cell B. So that just talks about that. So when we talk about surface area, the more the surface area of a membrane, the greater the diffusion rate. A large cell size is going to mean small surface area. And then also to take note, recall I talked about microvilli. Now looking at this cell A and cell B here, looking at cell A and cell B, which of them is going to be very effective which of them is going to be very effective in the spread of molecules? If particles are to move into the cell and particles are to move into the cell, which of them will the particles get in very quickly? Which of them will have a higher rate or a faster rate of diffusion? It's going to be cell A, simply because of the microvilli. What microvilli does is that it increases the surface for absorption. It increases the surface for absorption. This is just like a case of having a towel with numerous rough edges for absorption and having a plain white handkerchief for absorption. Of course, the towel is going to be more effective simply because it has what we call microvilli. So microvilli here increases the surface area for absorption. So what we are saying is that the more the surface area of a membrane, the greater the diffusion rate. And also take note, a large cell size is not going to have a large surface area. You see, large cell size, the larger a cell, the smaller its surface area. That's it. Another thing that determines the factor that affects the rate of diffusion is the distance. The distance. I said it decreases with distance. Look at the distance. Look at the distance here. It decreases rapidly with distance. As take note of this. This is a UTME question. It falls in proportion to the square of the distance. It falls in proportion to the square of the distance. Diffusion is only effective over very short distances. The shorter the distance, the smaller the cell size, the more effective the rate of diffusion, the more effective the cell. The thicker the membrane, the more the thickness of the membrane. If a membrane is thin like this and the membrane is this, let me make this a membrane very thick. The more the thickness of the membrane, that's what we're talking about here, the lower the diffusion rate. The more, if a membrane is thin, it means less distance to penetrate, the more effective diffusion. So they are faster the rate of diffusion. So what I'm saying here is that diffusion rate decreases rapidly with distance. It falls in proportion to the square of the distance. Diffusion is only effective over very short distances. And that's another factor that limits cell size. So we say a cell is small for effectiveness reasons. That a cell is small is an advantage. That a cell is large is not an advantage, because biological communication or diffusion of materials across the cell surface membrane to the center of that cell is going to definitely be slow. Now, these three factors we've talked about: the steepness or the difference in concentration gradient between the two points A and B, the surface area of the membrane, and of course, the thickness of the membrane, or what we call distance here, is summarized in what we call Fick's law of diffusion. Fick's law of diffusion. Fick's law states that the rate of diffusion is proportional to the surface area of membrane. That is, the more the surface area of membrane, the more or the faster the rate of diffusion. The more the steepness of the concentration gradient, the more the rate of diffusion. So you can see Fick's law states that the rate of diffusion is proportional to the surface area of membrane. It is proportional to the difference in concentration across the membrane, that's to the steepness of the concentration gradient, and it is inversely proportional to the thickness of the membrane. The thicker the membrane, the slower the diffusion rate. If we have a thin membrane, then the faster the rate of diffusion. So you can see rate of diffusion is directly proportional to the surface area. Rate of diffusion is directly proportional to the difference in concentration between 2 points. That's the steepness of the concentration of the diffusion gradient, and it is inversely proportional to the thickness of the membrane. Take note, it is inversely proportional to the square of the distance. So these are the three factors that affect the rate of diffusion, and then the following factors hasten that speeds the rate of diffusion when you eat. When there is an increase in temperature, imagine dropping a lump of cube sugar in hot water and in cold water. Which one is going to dissolve? Which one is going to spread very quickly? You see that the one in hot water is going to spread very quickly. So eating or increase in temperature, speeds up the rate of diffusion. And then when you stir or shake, that also spreads, makes a molecule spread very quickly, in thereby increasing speeding up the rate of diffusion. So that ends that. We've looked at factors that affect the rate of diffusion, and we've summarized that in what we call Fick's law of diffusion. And also we looked at factors that hasten up the rate of diffusion: eating and stirring. And so let us quickly look at processes that involves diffusion in living things. That's talking diffusion in living organisms. Digested and soluble food diffuses from the villi of the small intestine into the blood capillaries, and then from the blood capillaries into the blood proper, and from the blood it's mobilized into the cells of the body. Look at the diagram on your screen. You can see the small intestine, you can see the villi. Of course, it will not be possible, if food gets into the small intestine, if digested food gets into the small intestine and goes on and on till it gets to the anus. No, it doesn't make sense. At some point, digested and soluble food must be absorbed into blood capillaries. So it means from region of high concentration to the blood capillaries where it is in low concentration, so that from there the right materials, metabolites, get into the cells. So that's what we mean by saying digested and soluble food diffuses from the villi of the intestine into the blood capillaries. And once we have villi, in other words microvilli, then absorption rate is going to be fast, diffusion rate is going to be fast. So digested and soluble food in the small intestine from the duct move into the blood capillaries via diffusion. Exchange of carbon dioxide and oxygen during respiration is by diffusion. There is high concentration of oxygen on the outside, there is low concentration on my lungs. So what that implies is that oxygen is going to move from region of high concentration to my lungs where it's in low concentration, and carbon dioxide will move from my lungs to the outside via diffusion. If you talk amoeba, it is aquatic, and just like most unicellular organisms, you have oxygen dissolved in the water. So there is high concentration of oxygen in the water. It then moves into the cytoplasm of the unicellular organism via diffusion, and waste materials, of course, eliminated, leaving the cytoplasm into the water via diffusion. You can see amoeba and water is by diffusion. Then you look at the gills of fish. How does the fish take in, the dissolved oxygen in the water? It lowers the flow of the mouth. Water containing dissolved oxygen moves in. Then the oxygen, you see the gills is supplied excellently with blood. It has a good blood supply. If you open the upper column and you try to remove the gills of fish, you see that you're going to have blood stains all over your hands. That's simply because once the water containing oxygen had moved in to the mouth of the fish, the dissolved oxygen then diffuses from the water where it is in high concentration to the gills, and then from the gills into the blood via diffusion before the water is then let out via the upper column. Now, lungs and blood in mammals, yes, exchange is via diffusion. Also talking about maternal blood circulation and the fetal blood circulation, you see nutrients and gases diffuse from the mother's placenta to the fetus, and waste products from the fetus to the placenta via diffusion. So the right materials from the maternal blood circulation to the fetal blood circulation is via diffusion, and then waste from the fetus to the mother is via diffusion. Also, you recall that plants use carbon dioxide for photosynthesis. The plant is going to the leaf because it is the leaf that contains the green pigment, chlorophyll. And when you look at the transverse section of leaf, you see the substomatal air cavity. Okay, what I'm saying is that carbon dioxide moves into the leaves of plants via diffusion. Oxygen produced as waste products leaves via diffusion as well. Also, when you talk about saprophytic organisms like mucor, rhizopus, yeast, aspergillus, penicillium, fusarium, talking about saprophyte generally, they carry out what we call extracellular digestion, releasing enzymes into dead and decaying organic matter. So when saprophyte releases, let's say mucor for example, when it releases enzymes into dead and decaying organic material, that breaks down that food into soluble compounds. So how will the mucor or the rhizopus or the fungi then absorb that digested food? It is going to do that via diffusion. So this is just to look at a few cases of diffusion in living organisms, and with that, diffusion is very simple. Diffusion is a very simple process. Don't forget all the important things there. It is movement of molecules from region of high concentration to lower concentration, and it does not require cellular ATP. So that is the end of diffusion. Diffusion in living organism. Remember that at the start we talked about the phospholipid bilayer, and we talked about the fact that the lipid part of the membrane is hydrophobic, is non-polar, and does not allow movement of glucose, amino acids into the cell. So we then talk of needing a membrane protein and carriers across the cell surface membrane to help carry polar molecules into the cell, and that way that gives rise to what we call facilitated diffusion. Facilitated diffusion is just like diffusion as well. Molecules, particles have to move from region of high concentration to region of lower concentration. Cellular ATP is not required. That cellular energy is not required. The only difference now is that in the case of simple diffusion, molecules just move into the cells, get in between the spaces in the cell membrane and get into the cell on their own. But in the case of facilitated diffusion, you need a carrier. You need a protein carrier to move those molecules into the cell, simply because those molecules now are not allowed in by the lipid part of the cell membrane. Look at what I'm talking about. Facilitated diffusion is the transport of molecules and ions across a membrane by specific carriers. In this case protein, from a region of higher concentration to a region of lower concentration. Neither simple nor facilitated diffusion requires energy. Take for example glucose. Glucose normally cannot get into the cell because of the non-polar hydrophobic lipid of the membrane. So what it does is that it attaches to carriers on the cell membrane, which then carries it across and drops it on the inside of the cell. You recall, for simple diffusion, this is high concentration on the outside of the cell. This is the cell membrane, CM. You can see particles or molecules are moved from region of high concentration to region of lower concentration. That is simple diffusion. In this case, it is still the same thing, high concentration to low concentration, but this time we talk of a carrier because the lipid part of the membrane will not allow the glucose. But with the help of the carrier now, the molecules are being carried and they therefore can pass through the cell membrane. Look at it. The combined glucose plus its protein carrier are soluble in the lipid and can therefore diffuse through. So facilitated diffusion is a mediated transport system where protein carriers on the cell membrane now have to mediate, have to facilitate the movement of molecules from region of high concentration to region of low concentration. And as you can see on your screen, look at that diagram there. You can see the outside of the cell, you can see the inside of the cell, how carriers, how proteins, carry molecules across into the cell. So that ends diffusion. And the next thing now is now talk about osmosis, talking about movement of water from a hypotonic solution to a hypertonic solution until a state of isotonism is established. Now, talking about osmosis, osmosis is about movement of water molecules. Osmosis is about movement of water molecules. It deals with water molecules. Let's start osmosis with an experiment. As you can see in the diagram on your screen, you have more of a capillary tube there covered with a selectively permeable membrane. That could be a cellophane, that could be a pig's bladder, that could be a bladder. Then a strong salt solution, let's say 3% salt solution, is poured into the tube. It is then lowered into distilled water. Now, after some time, what we found out is that the level of the solution in the tube was rising as the salt solution was rising, while the level of the distilled water was decreasing. That is only showing one thing. What it's showing is more or less like water is moving from the beaker, the distilled water is moving from the beaker into the salt solution in the tube. And after appreciable time, you can see that the level of water in the beaker had dropped significantly, while that in the tube had risen significantly. What is demonstrated there is a case of osmosis, where the distilled water has a low concentration, or what we call hypotonic solution. The strong salt solution in the tube has what we call a high solute concentration, or what we call hypertonic solution. So we can say here that, or infer here that, water had moved from a region of low osmotic pressure, or we say from region of hypotonic solution to the inside of the tube, hypertonic solution, through a semi-permeable membrane, because the semi-permeable membrane, talking about the cellophane or the pig's bladder, there is what separates the salt solution and the water, the distilled water in the beaker. So water had moved from a region of low solute concentration in the beaker to a region of high solute concentration in the capillary tube there. So that alone gives the definition for osmosis as talking about movement of water molecules. As you can see on the board, I have two solutions here. A is a solution, B is a solution. Let this dot, let it be solute, and let this hyphen, let it be solvent, let it be water. So what happens is that when you look at solution A and solution B, solution A has few solute, where solution B has, a very high concentration of solute. So what that means is that we talk of low solute concentration here, and we talk of high solute concentration here. This, if we put the same amount of water in A and in B, the solute here is going to absorb most of the water, thereby leaving this one with a what we call low water potential or low water concentration. And when we pour the same amount of water here, since there are less number of solute to absorb the water, there is still going to be significant amount of water remaining in A. So we talk of what? High water potential or high water concentration. So that said, also, since the solute here has more tendency to absorb water, to draw more water, we say that they have what we call high osmotic pressure. You can see the more the solute, it means the more the propensity to absorb or to draw in water. And that is what you have about solution B. So if we have put the same amount of water in B as we have put in A, B is going to, the water we put in B is going to be absorbed by the solute there, because that is what solute does. They absorb water. But if we put the same amount of water in A, we are still going to have much water left, because there are less solute to absorb it. What that means is that this has a high osmotic pressure. Since this solution A now does not have enough solute to absorb so much water, we say it has what? Low osmotic pressure. It has low osmotic pressure to draw, to absorb water. So again, when we look at the net movement of water as water moving from A to B, we can again define osmosis as the movement of water molecules from a region of what? Low osmotic pressure to a region of high osmotic pressure through a semi-permeable membrane until equilibrium is reached. Also, worthy to note that the solution which loses water is said to be hypotonic. And that's what we have here. The solution which loses water is said to be hypotonic, and the solution which gains water, which absorbs water, is absorbing water because it has a more solute there. So we call that hypertonic. That is the solution that gains water. When water moves from A to B, a stage will be reached where the solute, the concentration in both solutions is going to be equal. You see, when a state of equilibrium is reached, that is, neither solution gains nor loses water, we say isotonic solution or isotonic state has been reached. When we mention the word isotonic, what that means is that the rate at which water moves from A to B is equal to the rate at which water moves from B to A. It doesn't mean that water had stopped moving between the two solutions. Water is still moving between the two solutions, but this time there is a balance. The rate at which water moves from A to solution B equals the rate at which water moves from B to solution A. So that said, also worthy to note that osmotic pressure of pure water is zero. Purely distilled water is zero. As you can see in that diagram again, in that distilled water that does not contain solute, in that beaker of distilled water, as you can see on your screen, that distilled water, what it means here is that the osmotic pressure is zero. Now, we had a strong salt solution, 3% salt solution in the tube, and then both solutions are now separated by a selectively permeable membrane, which can be cellophane or pig's bladder. What you then have there is, you have a situation whereby the level of water in the tube suddenly begins to rise. That in the distilled water, that in the beaker, the distilled water suddenly begins to drop. That is simple. It is because the distilled water is having osmotic pressure of zero. That's low osmotic pressure. Inside the tube, that is high osmotic pressure. Water therefore begins to move from region of low osmotic pressure to region of high osmotic pressure through a selectively permeable membrane, and that is why it keeps on rising there. So with this, that ends gives us the definition for osmosis. You can see it is going to always be movement of water from, use any of this term to any of this term, through a semi-permeable membrane until equilibrium is reached. So that ends osmosis. The next thing we are going to be looking at is going to be application. Yes, application. That's in the second part of this lecture. The next thing we'll be looking at applications of osmosis, talking about things like turgidity, plasmolysis, haemolysis, crenation, and the likes. We're going to be comparing osmosis and diffusion, and as well we're going to be looking at bulk transport, talking about endocytosis and exocytosis. Of course, we're going to look at experiment of osmosis. And so with that, we have come to the end of this lecture on movement in and out of cells. Not the entire lecture, though. That's just the first part of the lecture, where we've looked at diffusion, facilitated diffusion. We've looked at what the cell membrane looks like, and now we have been able to look at what osmosis is all about, talking about movement of water.